why we are studying about Human Genome Project. So before going on to the topic, let us first talk that what exactly this term meant for. Children, talking about the Human Genome Project, so it is something related with the human. Now the another important term in the subject is genome. So genome is an organism's complete set of DNA, right? किसी भी organism के complete set of DNA को हम लोग बोलते हैं genome, and it include all of its gene, right? So each genome contains all of the information needed to build and maintain that organism. Talking about the humans, a copy of the entire genome, like more than 3 billion DNA base pair, is contained in all the cells that have a nucleus, right? So when we talk about the genome, so the complete set of the DNA including all of its genome of the organism is termed as genome of the organism. In today's topic, we will actually discuss about the human genomes, right? Now coming to the topic that what is Human Genome Project and why we are studying about Human Genome Project. Human Genome Project is the project in which all the genes or the nucleotide sequences, the way nucleotide or base pair are present in the DNA of the human beings were sequenced in all of the chromosome, right? तो basically Human Genome Project बनाने का मकसद क्या था? Human Genome बनाने का मकसद था कि जितने भी human beings में न्यूक्लियोटाइड यानी कि बेस पेयर्स यानी कि जीन जो प्रेजेंट हैं उनके सीक्वेंसेस करना in all of the chromosomes, right? So sequencing of the gene is the main aim of the Human Genome Project. As we know, human beings have 23 pairs of chromosome in which there are several thousands of genes present and is made up of base pairs. So the aim of Human Genome Project is all these genes are sequenced, right? So the sequencing of these genes is the main aim of Human Genome Project. Now the question is that why Human Genome Project? So children, in each and every individual there are differences in gene sequences because all individuals have several differences in their physical as well as genome structure. For example, children, eye color, DNA fingerprints. So these are different and unique for each and every individual. That's why Human Genome Project means the sequences of human genome was very important and also to determine which gene code for which protein. Now children, this was started in 1990, right? So this was started in 1990 and estimated cost is around $9 billion US. And it is estimated that if sequences one gene required is around $3 US, then there are total 3 into 10 to the power 9. Then there are total 3 into 10 to the power 9 genes. So it was required $9 billion US. So the project completed in around 2003. So children, it took a very long time. As we can see, 13 years. Now why this Human Genome Project? Now why this project was called a mega project? इस project को mega project क्यों कहा गया? As I told, human genome have 3 into 10 to the power 9 genes. So children, if in a book there are 1000 pages, right? And in one page there are 1000 sequences. right? Gene sequences can be written. Then the completion of writing the sequences of human gene is required around 3,300 books, right? Like this. So from this only you can understand that how big the approach was to sequence all the genes in human beings. Now coming to the next important point, that is goals of human beings. That is goals of Human Genome Project. What are the goals? And one more point I want to add, that sequencing human genome was humanly not possible. एक humanly अगर ये काम हो जाए तो it is not possible. That is why it is done by using bioinformatics, right? Okay. Now coming to the goals, like the very first thing, to identify all the genes in human beings, that is from 20,000 to 25,000 genes in human beings, right? So that is the important goal. The next point is to determine the sequences, that is 3 billion chemical base pair present in the human DNA, right? It is again an important goal for that project. Thirdly, store this information in the database. Fourth is improve the tool of data analysis. Fifth one is transfer related technologies to other sectors such as industries, right? And sixth one is to address and deal with the ELSI, that is ethical, legal and social issues, right, that may arise from this project. In simple words, we can say that determine around all the genes 20 to 25,000, determine all the base pair in the sequences, base pair as well as they known adenine, guanine, cytosine, thymine, because there are 4 bases A, C, T, G, right, found in the human beings. Transfer related technologies, improvement in tool and technologies used in each project, and ethical, legal, social issues dealing of it. Now children, the question arises, who coordinated this project? So US Department of Energy and the National Institute of Health, both of US organization, and also other nations are involved such as China, Japan, France and Germany, right? So these are the agencies and nations are involved in this project. Now coming to the next important point, that is methods used in Human Genome Project. Still now we only deal with information of Human Genome Project. Now coming to the process. Process is actually used to sequence human genome. Basically 2 methods are applied. One is EST, that is expressed sequence tags. And the next process, sequence annotation. Now let us see what is express sequence tags. Children, in this method only the gene which code for or which synthesize RNA were sequenced. Now what is done in sequence annotation? First, the nucleotide, all the genes or all the nucleotides as well as base pair we can say, including both coding or non-coding sequences, right, were determined and identified, and then they were sequenced according to their function, means one after another. For example, lac operon. What we know about lac operon? There are 3 structural genes, that is Z, Y, A. So Z codes for beta galactosides, Y codes for permease, A codes for transacetylase. So Z is the main enzyme then come to the role of permease, then come to the role of transacetylase. So according to that it is used, right? Now coming to the vectors used, and the method used. So what is done? So from a cell, a DNA is isolated. Then the DNA was broken into the small pieces and inserted into the suitable host using vectors. And then they were amplified. So later sequences become easier because the amount of DNA means each small nucleotide was increased in amount. मतलब कि उसमें amplification हो गया, right? So amplification is there, the amount of DNA is increased. Amplification means a small DNA fragment increased in amount, right? Now coming to the which vector is used, right? कौन से vector use हुए? So two important vectors are used, that is BAC and YAC. BAC stands for bacterial artificial chromosome and YAC stands for yeast artificial chromosome. So these two type of vectors are most preferable. Now coming to the silent features of the Human Genome Project. So Human Genome Project has many silent features. First, they have astro studied. Also we must know how fragments were sequenced using automated DNA sequencer which was developed by Frederick Sanger. So fragments were sequenced using this machine, that is automated DNA sequencer. Then what to say? Alignment of sequences were not humanly possible, right? So the sequences की जो alignment होती है, it is not humanly possible. Therefore that was done by bioinformatics. That is one of the new area in the biology, right? So here it's given that this chromosome then coming to the DNA, then it is automatically sequenced. Now coming to the next part, as we know in human beings there are 4 autosomes present in the body cells and 2 allosomes, that is X chromosome and Y chromosomes are present. Now coming to the silent feature. In human genome there are 3164.7 million nucleotide base pairs, right? Now every gene consists of 3000 bases and it can be vary in size. For example, largest human genome is dystrophin that constitute around 2.4 million bases. So dystrophin is the largest human genome. ये बहुत important question है जो MCQ के form में आ सकता है, right? So dystrophin is the largest human genome और इस एक genome में ही जो है तकरीबन 2.4 million bases present होते हैं. Now total number of estimated gene that is thought by the scientist is 30,000. Previously, before starting of the project, it was thought that it is 80,000 to 1,40,000. Now functions of approximately or more than 50% of the discovered gene of still not known. And less than 2% of the genome codes for protein. And repeated sequences are very common. Means large portion of human genome constitute the repeated sequences. Now what are those repeated sequences? It don't have any direct function, but they shed light. They tells us about the chromosome structure, dynamics and evolution. Now chromosome 1 has most gene and sequencing of chromosome 1 was completed in around 2006. Right? Chromosome 1 has more gene, that is 2968. Right? And Y has fewest, that is 231. So you can just understand the difference in the ratio. Chromosome 1 has 2968 and Y has 2361, right? Now coming to the application and future challenges. Okay, now we know that human genome just revolutionize our life. It can increase our lifespan. It can make us disease free. then we can study everything about all the genes in our genome and in particular tissue, how number of DNA work together in connective network, or how the chemistry of life is there. So this all we can understand with the help of this Human Genome Project. So I hope you can understand what is the Human Genome Project. So in a very short form, when we talk about the Human Genome Project, so Human Genome Project is actually the project which is basically deals with the sequences of human genome. Genome is the set of genes, right? And children, this Human Genome Project was called a mega project, and it was first launched in 1990, right? And the scientists which work on this project, which started this project, which first sequenced the human gene, is SCARG. So it is the very important project. It is the very important project which actually deals with the sequences of human genes, right? And by that, all the genes in genome can be studied together. They can help us to understand how tens of the thousands of genes and proteins work together in the interconnected manner, and it also helps in the diagnosis and the treatment of the disease. So I hope that it is clear to you. So if you feel any kind of query, so children, feel free to ask. So till then, have a nice day and thank you.